Switching gears now, we'll look at a methodology, at a technique that was very popular. It was the thing to do as far as high throughput studies of sequence related work was concerned about 15 to 20 years ago. Uh, Somewhere about 10 years ago, it fell out of favor as next generation sequencing methodologies came to the fore. But I think as we begin to look at this high throughput methodologies, bioinformatics, kind of a large scale bioinformatics related to sequences, I think irrespective of whether you're going to use microarrays in your research, and you're likely not going to be able to use, they have not completely fallen out of favor, but most people don't use microarrays anymore. But microarrays were really critical to the field um, some time ago. And more importantly, I think microarrays, research related to microarrays, gave rise to biostatistics as an independent field of study. Okay, so it's very crucial. It's possible that in the near future, a problems or questions might arise which are potentially solvable using microarrays, and then we might be able to revisit a refined version of this methodology. So, microarray is the first example of high throughput studies, and they relied on hybridization. That is, the interaction between complementary DNA. If you put two DNA strands that are complementary, you're going to have a double stranded DNA. Okay, again, um, rationalized by hydrogen bonding, and you know what that is. So, with microarrays relied on hybridization, interactions between complementary DNA sequences that result in binding. Several hundred thousand simultaneous experiments could be carried out. You could do experiments for an entire tissue, and if the organism was small enough, you could do experiments related to an entire organism. And it could be used in there were several different ways in which microarrays found applicability, especially in health, because the assessment was related to it was a differential sort of analysis, and we'll look at this a little more. And it literally gave rise to biostatistics. So why did it give rise to biostatistics? Always remember that when you do experiments which are kind of on a large scale, okay, an ensemble of experiments, if you will, they are always governed by larger sample spaces, and larger sample spaces are governed by statistics. And very often people found that with microarrays, some of the experimental results, they were very popular. So a lot of the work got published in really good journals. But eventually they realized that some of these studies were done well, and people were just kind of breaking ground. So it's not really a criticism of the first researchers who use microarrays, but certainly what people found was a lot of the work was not reproducible. And again, that's because you do things in large scale, and people didn't pay attention to the statistics, to the quality of results, to things like standard deviation, to things like large scale, variances associated with large sample spaces. And biostatistics as an independent field of study came about to address some of this. So, a little bit about the technical aspects of microarrays. On a substrate, which could be a glass plate or something more specific, a polystyrene beads, for example, you attach short DNA sequences called oligonucleotides of a specific family, say, for example, olfactory receptors. Now, these short oligonucleotides that are attached on a substrate are called probes. Okay, so sample mRNA, identified, extracted, separated from a cell, converted to complementary DNA using reverse transcriptase, which basically converts RNA, which kind of reverses the process, reverses the transcription process, is then washed over the array. And the same can be done at the tissue level, an entire organism. Okay, so you could have 100,000 of these probes, and then you could extract mRNA, convert it to complementary DNA, and wash it over the probes. Whenever your cDNA found complementarity with the probes, they would form a double-stranded DNA, and you could associate the cDNA could be labeled with fluorescent dye, and when complementary is achieved, there is hybridization, double-stranded, and the strength of the hybridization tells you how complementary the two strands are, and this strength could be quantified using the fluorescence response. And then you wash the thing, the DNA, double-stranded DNA stays in place, and the fluorescence is captured as an image. Okay, and the intensity of the fluorescence is directly correlated with the strength of the binding of the doubles associated with the double-stranded DNA. So again, you see the fixed probes, and you have a label target sample, and here you have a full complementarity, and as soon as a complementarity arises, now you remember any A is going to bind to any T. That doesn't mean both strands are fully that full hybridization has taken place. So this is partial hybridization, and so you'll see the green stars are where full hybridization takes place, and you see the partial hybridization that takes place. And this is, for example, the substrate. And on these, you have the fixed probes. Okay, so there are various different kinds of experiments for this was used. You make a cDNA, reverse transcriptase. And so, for example, you would have to determine gene expression levels. For example, you could have a control, and you could have an experimental level, and then you could have different fluorescent dyes on each of those, and you push this over a this kind of a probe set. Okay, that is all the probes are on here, and then depending upon the intensities, you could have, for example, one wash for the control, and on the similar probe set you would take the experimental, and you would get both, and you would get the laser emission related to the fluorescent to the dye labeled DNA, and you would put these together, and then you would once you put the two on, exactly what probe is associated, because remember the probe sets are the same, so you know exactly what position is what. And then you would basically say, if a particular protein which hybridizes with a probe is that comes from your sample is upregulated, it would be red, and constitutive expression would mean that no change, because the green and the red would be the same. And for example, the level of intensity would give you an expression of the level of upregulation, or green would be downregulation. And you could look at it, for example, you could take, for example, a tissue which, from a healthy tissue, or you could take one where you would have a diseased tissue, for example, a tumor. And what you would do is, it's kind of you do this differential analysis, and you know exactly what protein, because you've already created the probes. So you know exactly which protein, because you know what's going to hybridize with what. And then you would say, okay, in this particular cancer, this protein is upregulated, this protein is downregulated, and this protein undergoes no change. So you really get if not a genomic view, certainly a tissue-wide view of cancer versus healthy. Okay, and that's why microarrays were so useful. And let's kind of do a wrap up on microarray analysis. So you have a gene selection, a small number of experiments result in large number of genes with varying expression levels. Some of the genes are housekeeping genes, not really important. In the sense, when I say not important, it means that they are not really overexpressed or underexpressed, or not really involved in the causation of a tumor. Okay. Other genes are of consequences. How does one determine which are important in the context of what is being studied, disease versus healthy, etc.? And one wants a smallest number of genes to give the best results for diagnostic purposes, because then you can really narrow down the proteins of interest associated with a particular. Um, Here is the microarray analysis steps. This is a very useful resource, and we will look at this resource, and you're going to learn something about resource called David. As a follow-up to our discussion on microarrays, we are now going to look at this resource called David. Um, it's an acronym for the Database for Annotation, Visualization, and Integrated Discovery, and it comes from the National Institute of Allergy and Infectious Disease, which is one of the institutions within NIH. And this is a really useful tool. It is actually a comprehensive knowledge resource that provides information related to genes, or essentially the products of a microarray experiment. And you can see that there are several features to this, and we are looking at version 6.8. So there are several features, and they undergo constant revision for more information. As more information is being produced. So there are 4 features: functional annotation, gene functional classification, gene ID conversion, and gene name batch viewer. So let's go into a quick description of what each of this is. Gene annotation enrichment analysis, functional annotation clustering, biocarta and KEGG pathway mapping, gene disease association, homolog match, ID translation, literature match, and more. The gene functional classification provides a rapid means to reduce large lists of genes into functionally related groups of genes to help unravel the biological content captured by high throughput technologies, microarrays being one of them. Gene ID conversion converts lists of gene ID accession to others of your choice with the most comprehensive gene ID mapping repository. The ambiguous accessions in the list can also be determined semi-automatically. And this is one of the discussions that are related to different people calling genes different things. But when you take all these synonyms associated with a gene and map it to a common identifier, if I have information stored in the resource as gene, for example, X, but then somebody says, well, I'm going to use a synonym Y. Let's say, for example, that gene was mapped to gene X. If somebody asks for gene Y, and we know it's a synonym for X, because it is mapped to because both X and Y are mapped to gene X, you can get the information for the correct gene. So that is what gene ID conversion means. And gene name batch viewer, display gene names for a given gene list, search functionally related genes within your list or not in your list, deep links to enriched detailed information. So let's look at one of these resources, and we don't have microarray experiment results, so let's go with kind of one demo list. Okay, you can also upload your own list, so that is saying that I got all these genes that arise out of my microarray experiment. Now I need to find out more about them. For example, I want to find out, have these genes been implicated in any disease? Do these genes belong to any biological pathway? I'd love to know more information about recent literature associated with these genes. And that's why this becomes quite a comprehensive knowledge resource. But so, for example, you can cut and paste a list. You can also upload a list that is a result of your microarray experiments. And then you can also choose, for example, different identifiers. Now, remember, I told you about this company that is, more than likely potentially out of business. Now, I'm not very sure about this, but maybe, but right now, Affymetrix used to be the be-all and end-all for a lot of this work. And you tell them what you want, and then they manufacture probe sets, and they made a lot of money leveraging the microarray technology. But now nobody hears of them. But anyway, so you see, for example, you could have gene IDs. We've looked at ensembles, so there's gene ID associated with ensemble, the MIR base, the mRNA GI accession number, ref-seq protein, all uniprot accession numbers, which is basically when we have our discussion on proteins and exphasy, we'll get some of this information. You could also go with not sure, in which case the resource will find, will look at your gene and try to map it to kind of known genes and see if that gene maps onto something that you weren't aware of in terms of kind of the ID or the type of ID. And it'll try and say, well, this looks like a GenBank ID, so let's kind of do our annotations, our knowledge extraction based on that. So there's different kind of IDs. We'll just go with the default right now. And so let's say, for example, if I go to gene list 1, and that has 15 unknowns, 149 Homo sapiens genes. So this is a result of some experiment, and that comes from the genome list 1. And you could, and that comes from the demo list 1. And if I can see what that gene list is, these are the gene lists. This is the genes that arose out of the Affymetrix experiment, and you can see that this is some format that is used to identify probe sets and genes associated with Affymetrix experiments. And immediately what you're going to get is, first of all, that they're all Homo sapiens, and we understood this. These are all the genes, the names of the genes that you get from here. And there was 150 odd genes. And you see here, you can click on related genes. Okay, so immediately, based on your experimental results, it knows exactly what those genes are. It tells you what that species is. And so let's click on related genes. So for example, for the genes of interest, the gene of interest, for example, the neural cell adhesion molecule 1, NCAM1, we clicked on related genes, and immediately you got the original one, which is a kappa, and kappa looks like some sort of a similarity metric. The color coding tells you that, and the kappa value tells you that you don't get a lot of very high similarities, but these are important. There is some similarity. They come from the same species, and the kappa value will tell you the relatedness. And then these are the genes that are of interest, that are similar to the genes of interest. Now you click on each of these genes, and you're going to get more information about these genes. It tells you what chromosome it comes from. It tells you what the cytoband is, a chromosomal location. It tells you what the ensemble gene is, and so you can go into that gene and ensemble, and what you're really going to look at is more information related to that gene. And we can go back a little bit here. So it tells you it could be the product of a pseudogene. Developmental stage PSPG are produced in high quantity during pregnancy. So immediately just from, and we're not even right now, we didn't even look at resources of genes that came from that particular experiment, but we've already begun to expand our knowledge base because we are now looking at these genes. Let's go back to our original list. And so we found out about related genes. Now let's look at that gene. So it tells you chromosome, it tells you chromosomal location, it gives you the ensemble gene ID. And I suspect if you probe this a little more, you could actually get into the location in the genome browser. Okay, and we're not going to do that right now. But what is all of this? All of these are the titles of papers associated with these genes. Now, it could stand to be a little more organized, but you can see a lot of work has been done. The gene symbol is NCAM1. It tells you something about the function. And if I click on one of these, as I said, it is the title of the article. And once again, you see how, for example, that gives you more information related to the abstract. If you have a link to a full text article, you can go and look at the full text article. Go back a little bit, and you'll see that it has already clustered and categorized the results that you had into the different, and from these 149 genes, you realize that it found specific protein domains. It found nothing in terms of protein-protein interactions, tissue expressions. It found related to one disease. It found some functional categories. And let's look at the functional annotation clustering. So what you'll see here is that based on functional annotation, it tells you what the go term is. It tells you what the p-value is, and so these are some statistical numbers, and so you can think in terms of the function associated with the genes of interest. Okay, so this is all 150 of these genes. You begin to find all the functions associated with these genes. If I click on disease, for example, it found that there's some information related to disease, and so the things in red are the default. So I can click on this, and I can look at a chart related to the disease, and it tells me that a lot of this is associated with gut disease, HIV, lung cancer. Again, related terms. If I click on this, it'll tell me the related terms to the disease of interest. And okay, it'll tell me what the functional categories are based on the ontologies, sequence features, etc. I can click on this bar, and that'll give me the same information as I got from the chart. So it tells me the sequence features. There's a binding site, there's a chain, a 5-hydroxy receptor, related gene. And again, I don't know anything. I do not know anything about this work, but I'm just pointing out the various features, because people who are doing microarray experiments come with a lot of contextual knowledge of the information that they have, and so, as in terms of the specifics of the knowledge, this information that is being provided is actually really useful for people who are working in that particular field. Okay, so let's look for one more thing, that is pathways and the various pathways. And so if you click on this resource for pathways, we can see that that these genes are found in these particular pathways. So let's click on tryptophan metabolism, and it takes us to a related KEGG resource. So what it has done is it has taken the KEGG pathway, and so what you see is, you might see a lot of these are not lighted, for example. That is, so when KEGG creates its pathways, it creates its pathways using kind of all the genes associated for all the species. Okay, and then the ones that are green in color are associated with proteins or enzymes or DNA or other molecules that are involved in that pathway for that particular species. And so the gene of interest that we are looking at has already been kind of is pointed out here. Now, we can find out more about that gene just by mousing over, and that's 3-hydroxy anthranilate 3,4-dioxygenase, the HAAO. Now, if I click on this, it'll give me more information about that protein. Now, about that particular gene. Now, one thing we can also do is, this tells us something about the KEGG pathway. But one of the other things I can do is I can go to the Kyoto Encyclopedia of Genes and Genome, and I can look for, so we look at the tryptophan metabolism, and this is the reference pathway. As I said, it's got everything associated with all the species. But if we look for a specific species, then it'll only highlight the ones of interest. Now, if you go back to our pathway analysis, the one with the star was 113.11.6 enzyme, and this is written in the enzyme nomenclature. So 113.11.6. So 113.11.6, this is the resource that we looked at. So in KEGG, it'll give you a different set of information. So it'll tell you about the gene name, this various synonyms for the genes, the full name for these genes. It's involved in the tryptophan metabolism. It is also involved in other metabolic pathways. And it tells you here's the, I'm sorry, it was not 113, it was 1.13.11.16. And it'll tell you what the enzyme nomenclature is and why it has gotten that name, and that is it's an oxidoreductase. That's what the 1 stands for, and the 13 means it's acting on single donors of molecular oxygen, and the 0.11, the subcategory, is incorporation of 2 atoms of oxygen, and that 0.6 is that's a very specific gene, and it'll give you a sense of how this is. Now you'll get an information related to the orthologs related to that particular gene. Okay, you'll get information related to the paralogs related to the gene. It tells you in PFAM, which is a protein families resource, exactly what kind of binding regions or what kind of motifs are associated with what kind of because of various functional structural features, what kind of families it might belong to. It tells you all the IDs. If you went to the gene ID, the protein ID, this is the information related to the gene, what that ensemble information is, Vega, which is something we didn't discuss, but it's another genome browser, what the UniProt ID is, and we'll look at some of these resources some other time. It tells you LinkDB. It tells you there are 3 PDB structures associated with this gene, and this is JMOL. This is a Java-based app which allows you to dynamically manipulate and look at the various facets of the structure of the protein on the web. It tells you what the amino acid sequence is. It tells you what the nucleotide sequence is, and so you can see that you can start out with the pathway in David, and that'll give you some information. As you can see, then that's the one that's necessarily highlighted. The list of genes are shown in red. Let's go back and see if we can get any kinds of information. We look at protein domains. So for example, the various resources which give you information related to the protein domains that are involved in this list of genes. So now we're not focused on the genes. So this list of genes covers what kind of protein domains. So then you might say, well, and this is a little bit, you might think this is a little bit far-fetched, but if you say if there is a commonality related to the protein domains associated with all these 149 odd genes, if that's the case, then maybe there is some sort of a correlation between, for example, the disease that we are studying, or some function that we are studying related to this set of microarray experiments. And is there a correlation between the protein domain associated with that? And so you don't find any protein interactions, you don't find any tissue interactions, and this is important. This is important to understand. Okay, so this is a little bit. It gives you a little bit of an overview of David. Once again, I can point to the features, but I suspect that if you kind of had some context of an experiment that you were involved in, and you knew exactly why you wanted microarray data, and you knew exactly what, or you had a sense of what was going to come out and what in that list of genes you were looking for, you would find this of very specific interest, and you can kind of look at the various charts that are available, and this is a very, very comprehensive resource and a very useful resource. Okay,